Welcome to Pharmaceutical Technologies Podcast, Challenges with Extractables and Leachables in Medical Devices. This podcast is brought to you by Waters Corporation, the world's leading specialty measurement company. To find out more, please go to www.waters.com. And now, here's your host for this podcast, the contributing editor for Pharmaceutical Technology, Cindy Dubin.
Hello, everyone. This is Cindy Dubin, contributing editor for Pharmaceutical Technology, and I'm here with Dr. Andrew Fielden, E&L European Strategic Director at Hall Analytical, and Dr. Ben McCreath, Strategic Program Manager for Waters Chemical and Material Business. Hey, Andrew and Ben, thank you for being here today.
Hi, Cindy. Thank you for the chance to speak to you all today on this podcast.
Yeah, thank you, Cindy. Pleasure to be here.
So, kind of jumping into it here, Ben, chemicals of concern can come into contact with people through a multitude of ways: food, medicine, cosmetics, personal care products, even life saving medical devices. To understand these chemicals of concern, different extractable and leachable standards and regulations exist for a range of the market. What would you say the main challenges are from moving from one E&L related standard to another, such as food to pharma to medical devices, or even moving between food contact materials, pharma, and medical devices?
Yeah, so we hear this being asked a lot. So being an analytical solutions vendor, it's mainly within the context of, will my existing instrument work for this? And indeed, once your analyte is in the vial being presented to the LCMS instrument, for example, there's a lot of overlap in the analytical workflows. So for the most part, your LCMS will be suitable for the required activities, but there's much broader considerations with respect to the sample preparation and analytical thresholds that Andrew's probably better speaking to.
Thanks, Ben. And yeah, it's correct that once you have a solution for the analysis, whether it be for food contact, medical devices, or pharma, the same analytical techniques, the same analytical approaches will take place. The only significant differences are around the limits required based on the route of administration or use, but also the solvents required, whether it be for food contact or pharmaceuticals, the levels, the associated risks with a given product. So a lot of the differences around the guidances are purely in sample preparation to hit the required levels. And as Ben said, once you have the solution, the analytical techniques are the same. The solution doesn't know if it's for food contact or pharma or medical.
You mentioned guidance, and just last year there was a major revision to the E&L guidance for medical devices captured in ISO 10993 Part 18. Can you please share with the audience what the major challenges posed by the update include?
Okay. There was the significant change in January of 2020. It went from a document that was quite vague in terms of the chemical characterization, the E&L activities required, to something that was a lot more thorough. It expanded up to over 60 pages and brought in the key concept of the analytical evaluation threshold, which gave you guidance and thought processes of how low to go. At what point do you stop the analytical testing, because any chemical that you detect below the threshold is deemed to have negligible safety concerns. So the changes were quite stratospheric in that terms, that the bringing the AET, mentioning the choice of solvents, so multiple solvents now, and the range of analytical techniques, marrying it up a lot closer to the guidances around pharmaceutical products, so the likes of USP 1663 and 1664.
And it does seem that the calculation of the analytical evaluation threshold is a key aspect within the recent changes. Is it reasonable then to use a one dose based threshold or a safety concern threshold for the primary packaging and then another for the device?
As the AET is an identification threshold, and it's used to develop the limits required for an leachable or an extractable study. If the two parts were related and overall the same product would be given to a patient or an end user, then the same AET should apply using the same dose-based threshold, the DBT or the SCT. So whichever one you picked, probably the lowest, will be the one that will be used to derive the AET.
Okay, and when performing identification and quantification of extractables and leachables, what is the best approach in deciding which standard to use as a comparison once an analytical evaluation threshold has been calculated?
This is probably one of the most challenging parts of the extractable and leachable testing. Every chemical species has a range of different physical chemical properties, ranging from molecular weight, log D, boiling point, vapor pressure, is it an acid, is it a base, a whole raft of things that can actually be around a given chemical species. So you need to have chemicals that are a range of these to help derive a particular standard for the AET. So you need to take into account all those factors and probably have multiple standards that can achieve different aspects around the AET for different analytical techniques, whether it be for GC, LC, headspace, or whatever techniques required.
And are there any other major considerations regarding the analytical setup to consider that impact regulatory compliance?
So there are a few levels to this, but reason dictates to start by looking for a solution that will allow compliance to be achieved. So ensure you have data integrity. So deploy informatics that conform to 21 CFR Part 11, for example. So you need to ensure that your system and your software can be qualified and validated, and make sure you have a robust solution with traceability and transparency.
And following on from Ben's answer to that, in terms of practicalities, a lot of the challenges are around limits of detection, devising the right study so you can achieve the analytical levels required based on the AET, understanding your equipment, what level it can achieve, so whether it can achieve 1 microgram per mil, 1 nanogram per mil. You can derive and develop your study of how many samples, how much solvent do you need to enable you to achieve the right analytical setup.
So, you both know that the term medical devices cover a vast range of products. In your experiences, are there any particular dosage forms or devices that are particularly challenging to test?
Most challenging devices are those that are either very large in size, so require a large solvent volume, which then dictates a high amount of solvent, which gives you a very low detection level, making subsequent analytical workups, whether it be concentration steps or increase the amount you inject with the analysis, more challenging, as well as those that are absorbed by the body that require complete dissolution, and having dissolved normally some very complex formulations or devices, which makes the analytical testing far more challenging.
Similarly, there are a large variety of materials used in designing and manufacturing medical devices. Are there particular materials that we should watch out for that may have a higher extractables or leachables risk profile?
In general, there are no materials that are completely risk-free, but the more amorphous the material is, the more likely or easier it is for a chemical species, an extractable, leachable, to migrate through the material and out into the product. So in general, elastomers are more concern than plastics, but also glass and metal are not risk-free. So you'll see more species typically from elastomers than you will from the plastics, but you'll quite often see maybe aspects around glass delamination or typically for metals, surfactants or cleaners that during their manufacture may still be present, but may come off into the product and may affect the patient.
Okay.
Yeah, and as I'm just saying, that I mean there are variations, and another important area is the non-intentionally added substances. So, for example, thermosets, you're more likely to see those, which gives rise to potentially more elucidation. But to me, the important bit here is the interaction between the supply chain as well. So technologies such as LCMS can detect many different compounds, and using the elucidation tools we have, you can go through the process of identifying the multitude of peaks. But by far the quickest route is to have information from the supply chain as to what could be in there. And then you use this to build a library of compounds for your LCMS. So you can then target these compounds, and the screening process becomes so much simpler and a lot less time intensive.
Patient safety is, of course, paramount when considering medical devices. And with contamination being a major potential risk, disposable and single use systems are very common in the industry. What are some of the challenges with introducing more sustainability into medical device products?
Yeah, so the footprint of the medical device industry is substantial. So trying to improve the sustainability whilst reducing costs and yet having no negative impact on the patient safety. So I mean, one can quite easily see why this is such a challenge. So some of the research that I see having a positive impact comes from the supply chain again. So provide a material that is sourced from sustainable resources, for instance. So intuitively, it seems easier to work from the monomer level rather than nature's polymers. But they do say that innovation can be accelerated through constraints. So trying to replicate the performance properties required with an inherently more heterogeneous source, whilst maintaining the acceptable E&L profile. So I just look forward to seeing some of the outcomes here.
And I'd agree with Ben on a lot of the points that it is challenging at the minute, considering the input raw materials. It's much easier to get a virgin raw material and use that to achieve the highest quality standards required for medical devices, pharmaceuticals, the food contact compliance. There are a number of recalls for various issues. Kellogg's had 28 million boxes of cereal recalled a number of years ago due to the use of recycled cardboard. But a lot of the polymer manufacturers are trying to strive to use different additive packages to achieve the ability to go back to the monomer, as Ben's discussed, and just go back to that to enable you to polymerize it in a very clean and very pure form.
So, before we let you go today, is there anything else you would like to share with the audience regarding medical devices and the regulations that govern them?
I think in terms of the medical devices, we will still wait to see the ISO 10993 Part 18 standard bedding in. It's just been just over a year now that it's been out and being fully available. We've seen some comments by some regulatory authorities, notably the FDA, on bits that they accept and necessarily don't accept. But we'll now also have to wait and see what happens with the proposed new standard, the ICH Q3E for extractables, and see what that holds in the future.
Yeah, it's certainly a very interesting area at the moment. There's a lot of activity in the space and obviously more to come, as Andrew's just articulated. So, I mean, the bits that I see that are really key are these interactions up and down the value chain. So ensuring that the critical information is shared to help ensure that basically nothing's missed and that characterization is streamlined for what can be it's not the most facile of analysis to perform.
Andrew and Ben, this has been really interesting to learn about extractables and leachables in medical devices. I thank you so much for your time today.
Thank you. You're welcome. It's been a pleasure speaking.
Lovely to speak to you, Cindy. Thank you.
This has been Cindy Dubin, contributing editor for Pharmaceutical Technology. Thanks to all for listening.
You've been listening to Pharmaceutical Technologies Podcast, Challenges with Extractables and Leachables in Medical Devices. This podcast is brought to you by Waters Corporation, the world's leading specialty measurement company. To find out more, please go to www.waters.com.